This is the do-it-yourself DNA experiment. In front of you we have all the materials needed to conduct the experiment. So we've got the measuring spoons, measuring cups, baggie, cheesecloth, salt, funnel, we've got toothpicks, rubbing alcohol, various cups, and the strawberries. So let's get started.
So as many of you know, experiments typically follow a 5-step scientific method where you have the kids asking a question, stating their hypothesis, then conducting an experiment, analyzing the results, and then forming a conclusion. For our do-it-yourself DNA, I came up with the question, do strawberries contain DNA? And if so, how can we tell? So if I had to make an hypothesis, I would say that I think strawberries have DNA, and we will be able to tell this by the different chemicals we throw into our experiments. Usually DNA is very hard to see with the eyes, but I think this experiment will simulate how people are able to take DNA samples by the chemicals, which will make the DNA come together so that it is visible to the eye. All right, now we will start with our experiment.
Okay, now it is time to start the experiment. So, in this cup to the right, we have a mixture of water, a third cup of water, a half teaspoon salt, and 1 tablespoon of dishwashing liquid. Here in the left side glass, there is the cheesecloth is placed on the funnel, and so that it covers the entire funnel. We will set both of these aside for now and grab our baggie for the crushing of the strawberries. Performing the next step in the scientific experiment, you might want to ask your kids, how do you think the detergent and salt will affect the strawberry cells? So we're going to take this, 3 tablespoons of this, and put it into our bag. So you might want to just question them. It's a pretty open-ended question, so that's good. Okay. So 3 tablespoons of the liquid we made prior. So, as directed, we will zip the baggie. And see what starts to happen. Next, we're going to pour the mushed and mashed strawberries into the funnel and strain it using the cheesecloth. One question you could pose here is, how does the filtered strawberry liquid look? That is the liquid that has been, or the mashed strawberry part that is in this part of the glass now that we'd be analyzing. So give it a few minutes to really get strained, and we'll come and take a look at it in a few minutes. All right, so now we've got our strained mashed strawberries, and we're going to use that juice to pour about 1/4 of, usually you would have a small jar. Sadly, I have only this, so we're just going to pour about, you would pour about 1/4 of a cup in a small, like, baby food jar, but we're just going to try this route first. You might not end up using the whole thing of strawberry juice, just 1/4 of the way. Okay, and then you want to add about 1/2 of a cup of cold rubbing alcohol so that it's about 1 inch of a layer. Okay, so you do not want these two elements to mix here. You want to, you'll start to see a gooey, clear, white, and maybe stringy type of stuff forming in between. So you might, and while you're waiting for this, you know, you've asked, we strained the strawberries. You know, kids can discuss how does this filtered strawberry liquid look. That can be discussed while you're waiting, because that did take me a few minutes. So just to make the waiting process a little less tedious. And then you've got this now. And you might want to ask, do you see anything in the jar that might be strawberry DNA? If so, where in the jar is it? So this, if you got a closer look, is developing that stringy, gooey, clear white material in the middle there. We're going to further explore this. We are going to take a toothpick or a skewer and look at the point in which the two layers meet, the rubbing alcohol and the strawberry mixture. It's gooey, it's clear. Everything the experiment kind of informs you about. So did you see anything, this was a question you might ask your students, did you see anything stick to the skewer that might be DNA? Can you spool any DNA on the skewer? So here we did get some pretty good amounts of DNA, and that's how, it's kind of a simulation as to how people get swabs of DNA, or if they need to obtain some kind of DNA, that is kind of how it's done. But now we're going to go into what happened in this experiment. You might be wondering what happened in our DNA experiment. When we added the salt and detergent to the smashed strawberries, the detergent helped to pop open the strawberry cells. This caused the cells to release their DNA into the liquid. At the same time, the salt helped create an environment where the strands of DNA could gather together and clump, making it easier for us to see the DNA. So when we added the salt and the detergent mixture, you might have seen a lot of bubbles, but this was also happening while we had put the detergent and salt mixture into the baggie. So when we added the cold rubbing alcohol to the filtered strawberry liquid, the alcohol should have made the DNA come together and separate itself from the rest of the liquid. You should have seen the clear, white, gooey DNA strands in the alcohol layer, as well as in between the two layers. It's kind of clumping together now. A single strand of DNA is extremely tiny for the eye to actually see, but because the DNA clumped together in this activity using the products that we did, we were able to see what is called octoploids. Octoploids means something has 8 genomes, or it's a set of genes. So we were able to see those using the household chemicals that we did. So there you go. So in conclusion, we were able to determine that strawberries do have DNA. After analyzing and questioning and putting together all our work, this is what we found. And we also discovered where we found the DNA and what it looked like, and
the clear, gooey mixture that was inside the 2 layers. So one thing that I would do differently for next time in this do-it-yourself DNA experiment would be to have some sort of maybe controlled group or variable groups. It was, if anything, if we did science groups, the science, each group would have essentially the same result and outcome, and maybe having some variables would, like a variable group or 2 would kind of change things up and get some variety, and that would be something I would do differently. I think the steps would be done with the kids throughout as the teacher models, the students follow. But again, maybe that isn't so inquiry based. But personally, at this stage, I don't feel as comfortable with inquiry based. I love the idea of it, but I'm I'm working on it. But that would be something that I would do differently. Is definitely get those variable groups in there so that there's variety among the groups. And other than that, this experiment can be found on a website called sciencebuddies.org. I will copy and paste the link as well so that everyone can access that. You do have to fill in some information about yourself, and you do get emails, but in order to see the instructions part of the experiments, you do have to sign up. But it's free, so just so everyone knows. All right, well, thank you for watching my experiment.